Okay, this is cells part 2, where we are looking at specifically eukaryotic cells with all of their organelles, but even more specifically, we're going to be looking at those organelles that are involved in a hugely important job, and that's making proteins. Please remember that proteins do everything in your body. They do everything. When you look in the mirror in the morning, you're seeing protein and the effects of protein activity. That's what made you. So please do remember, as we already have covered in the last podcast, there are two major types of cells: eukaryotic cells and prokaryotic cells. Prokaryotic cells don't have any of the things, or at least not most of the things that we are going to talk about today. They don't have all these membrane-bound organelles, these little compartments. Eukaryotes do, and you can see that they are much more complex. Please remember, we've already talked about this, why organelles. They allow for compartmentalization in cells, the compartmentalizing processes that are not compatible, so that different things can happen inside the same cell and not conflict with each other. We are going to be looking at different big jobs that cells do over the course of the next couple of podcasts. This is the one we're dealing with right now, and arguably the most complex, in that there are the most parts to this. So building proteins. Remember, proteins do all the work. Okay. Now, in order to build proteins, you've got basically a 4-step process that has to happen. You have the instructions for building all the proteins of your body, which basically means building you, of course, are in your DNA. So those instructions have to be read somehow. Then once we know the instructions and how to build the proteins we need to build them. But once we build them, they're not finished. They're not perfect yet. They have to be processed. They have to be modified a bit to do finally be fully functional proteins. So we'll look at how the processing of the proteins happens. And then finally, you have to get a protein to where it needs to go to do its job. It may not have to go anywhere. It may do its job within the cell that created it, but it may also be kind of chucked out of the cell and sent somewhere else in the body to do its job. So we need to look at how they get to where they're going. Okay, so four parts to building functional proteins. Okay, the organelles involved in all of this are several. The nucleus, of course, the home of the DNA, ribosomes, the protein factories that build the proteins, endoplasmic reticulum that moves protein throughout a cell, the Golgi apparatus that is going to polish the protein and make it perfect, and then little vesicles that kind of help the protein get to where it's going to go, ultimately sometimes outside of the cell. So this is the rough trajectory you want to keep in mind as we talk about this. This isn't perfect, but it's this general nucleus to ribosome to ER to Golgi. Vesicles kind of fit in a couple of different places, but that's the trajectory we want to think of. Okay, so let's start with the nucleus. Of course, we know the nucleus is the home of the DNA, the instructions. So basically its job is to protect that DNA. And while it's protecting that DNA, and that's very important, it can't be completely sealed off from everything else. Things do have to get in and out. So that's where the nuclear envelope comes in. So the nucleus, it's the structure of the nucleus is basically the nuclear envelope with all the DNA then packed inside. It's a double membrane, and that membrane has little pores. If you look here, here's that nuclear envelope, and you can see it's got little pores where things could go in or go out. Okay, and that's really important. Do remember that one of the things that has to get out is mRNA. You should have vaguely remember this from 9th grade. Remember, DNA can never leave the nucleus, so the DNA double helix is prisoner in the nucleus of a eukaryotic cell, but mRNA is created to be a copy of the DNA instructions, and that mRNA can leave and go out into the cytoplasm, where it will be read by ribosomes, more on that in a minute, to create a protein. Okay? Now, inside the nucleus, you often will find a structure that is so big and round, and here it is in a microscope view right here, that you would think it is an organelle. In fact, the nucleolus is really just a super highly active area of DNA, of the chromosomes, in which a lot of activity is going on. And what's happening is the ribosomes are being built there. The DNA instructions in this region code for ribosomal RNA and ribosomal proteins, and the RNA molecules are being made in this region. It is just a highly active region of DNA. It's all it is. It's not an organelle. It's not a membrane-bound compartment. Now, since the ribosomes are being made here, that's another reason why we need these pores, because the ribosomes have to leave the nucleus. They can't stay there and do their job. They have to get out to the cytoplasm. So you may see more than one nucleolus in a cell. It just depends on the kind of cell, how many ribosomes it needs. The more ribosomes it needs, the more of a more visible nucleoli you might see. Okay, so ribosomes. Please just remember, these are the protein factories. Their job is to read the mRNA and build proteins. So if I'm an mRNA strand, I'm going to thread myself right between the two parts of the ribosome, and the ribosome is going to just read the DNA instructions in the form of the nucleotide sequence that's on that. More about that later. Ribosomes, that we often call them organelles, are not organelles in the truest sense of the word, whereas remember, organelles are compartments. They're hollow. They're membrane enclosed compartments. Ribosomes are globs of protein and RNA, specifically ribosomal or rRNA. Okay, and if you were to look at the molecular structure of them, they would look like these globs, and it's protein and RNA combined. It's a solid glob that reads mRNA. That's its job, and when it reads that RNA, it makes proteins. It just spits out a protein. And builds it. Okay, so ribosomes are hugely important. The more proteins a cell makes, the more ribosomes it needs. As we are talking about this, think about structure and function. If you saw a cell that had lots and lots of ribosomes, you'd know that it had a job to make lots and lots of protein. All cells make protein, but some cells need to make more than others depending on what their job is. Now, there are two types of ribosomes, based not because they're structurally different, but based on where they are found in the cell. Some ribosomes are free ribosomes, and they're just floating in the cytoplasm, in the cytosol. Their job is to make proteins that are going to stay in the cell. They're not going to leave the cell. They're going to do something the cell itself needs done. But there are other ribosomes that are stuck or bound to a membrane, specifically the endoplasmic reticulum, and that's being shown here. The endoplasmic reticulum is this folded membranous sac, and on it, these little blue dots represent ribosomes. Ribosomes that are attached to the ER, their job is also to make protein, but these proteins are destined to be exported, to deliver outside of the cell in which they were made. Okay, so free versus bound ribosomes. So we've already mentioned the ER, the endoplasmic reticulum. Its structure, let me skip to its structure first. A hollow membrane that is just highly folded, but it is hollow. There's space inside this membrane. Okay. and it's folded around the nucleus. So this would be the nucleus here. So you can see the ER gets folded around it, okay? Its job is, the jobs of the ER are many, okay? And it depends, there's a couple of different types of ER. They create cell membrane itself. They can create and break down different compounds. They process and transport proteins. So lots of different things going on. So let's try to break that down a little bit. So types of ER. What we are talking about, of course, are rough ER and smooth ER. And this slide is trying to show you their arrangement. This is the nucleus here. The rough ER is most closely bound to the nucleus, but as you get further away from the nucleus, you see the ER loses its ribosomes and it's more smooth. So it's smooth ER. These two types of ER look similar but do different important jobs. Let's first talk about smooth ER and get that out of the way, because this really does not have a whole lot to do with protein, and we want to get back to our protein discussion. Smooth ER is very important. Without it, we wouldn't have cell membranes. The smooth ER makes phospholipids that builds membranes for the outer cell membrane, but also for all the organelles that need phospholipids to build their own membranes. It's built by the smooth ER, okay? The smooth ER will just pinch off and transfer phospholipid to other regions of the cell that need to build membrane, okay? The other things that smooth ER does, it has a highly specialized function in muscle cells that has to do with causing the muscles to contract. The smooth ER can store calcium ions or other kinds of ions as well, but in muscle cells it's calcium, that are critical. Their storage and release of these ions causes muscle contraction. So without smooth ER, your muscles would not be able to do what they're supposed to do. Metabolic processes. Metabolic processes just means making things and breaking things down, okay? We've already talked about how they make lipids. What we haven't talked about is how they break things down. In your liver, remember we talked about that carbohydrate glycogen that you have about a day's supply of in your liver. The smooth ER in the cells of your liver will break down that glycogen and give you glucose. So if this is a glycogen molecule in your liver, hydrolysis would occur and break that down, and you would get a bit of glucose from that. It's also in your liver a huge detoxifying organelle. It will detoxify poisons, bad things in your body, and make it so that your body can get rid of them. Alcohol is processed in the liver and detoxified by the liver. So really important, broken down. About that detoxification, I don't think you would need to know this, but just be aware, it's not magic. The smooth ER will add hydroxyl groups to different toxins, and those hydroxyl groups make drugs and other certain things more soluble, more dissolvable, and easier to just flush out of the body, okay? And it's interesting, if you are an alcoholic or you're drinking a lot, taking a lot of drugs, barbiturates, things like that, the cells, to compensate for all this poison they're getting exposed to, will create more and more smooth ER to try to deal with that toxin. And interestingly, once you've got more and more smooth ER to detoxify, that increases your tolerance to these drugs, so you have to take more and more of them to get the same effects. So, and that can even work against you with useful drugs as well. So just an aside, this is kind of interesting. Now let's get back away from smooth ER and back to our discussion of protein. Rough ER, it is where the ribosomes that are going to make proteins that need to be secreted out of the cell, it's where they sit. So they're bound to the rough ER. And if this is a little ribosome here, he's reading his mRNA molecule. What he's doing is he's producing a protein, and squirting it into the hollow, rough endoplasmic reticulum membrane. And then once he's done, that little protein will start traveling through to other places by traveling through the ER on its journey to try to get out of the cell. Okay. Interestingly, of course, cells that secrete a lot of protein are going to have a lot of rough ER and a lot of ribosomes making a lot of protein. Right here, what you see is a ribosome on the rough ER. There's its polypeptide chain. That protein gets a little bit modified in the ER. Little sugar chains will be added to it. And once that happens, this is called a glyco. Glyco refers to any carbohydrate, so these little bits of sugar that get stuck to it. And then protein for the protein. We'll talk about what these little sugar molecules do for it in a minute. But once it has gotten to the end of its journey in the ER will literally pinch off with its little protein, its little glycoprotein. And this thing that pinches off is called a vesicle, a transport vesicle. And this will get taken to another organelle for further processing of that protein. Okay? Real quick, going back to the fact that proteins, when they are in the rough ER, get a carbohydrate stuck to them. Okay, this carbohydrate add-on is just a short chain of sugars. If you ever see the term oligosaccharide, we know polysaccharides mean many saccharides. Oligo means a few. Okay, so a little short chain of sugars. It results in a protein-carb combination called glycoprotein. I already mentioned that. Often these things just get embedded in cell membranes, and they are used as cell identification markers to tell a skin cell from a liver cell or your cells from someone else's cells. And this obviously will have a very important function in the immune system. If you don't have the right ID markers, your immune system will attack a cell that doesn't have the right ID markers. So, at any rate, the Golgi apparatus is usually where one of these, if I'm going to go back here, this little transport vesicle that pinches off the ER is headed. Will head to the Golgi apparatus. The structure is a hollow stack of membranes. You need to think of it like a little stack of pancakes. And its function is to receive proteins from the endoplasmic reticulum and then finish them off, polish them up, sort them out, figure out where they need to go, and then ship them to the appropriate place. This is secretion. Secretion is sending useful products, proteins, outside of the cell. So vesicles play a large role. Vesicles deliver proteins to the ER, and vesicles deliver proteins away from the ER. So vesicles are hugely important. Within these stacks of ER, the proteins get modified and sorted out as they're passing through. Okay? Right here, you can kind of get this idea. We see that on the rough ER, we've got this little protein with a little bit of carbohydrate that gets added. It gets transported from the ER in a transport vesicle to the Golgi. The near or cis face is where it first lands, and then it passes through and gets modified. You can see the protein, see there? The little polysaccharide or oligosaccharide is getting modified as it passes through, and finally, when it's all done being modified, a vesicle pinches off, carrying the protein and budding, attaching to the plasma membrane and just kind of pushing the usable protein either out, or maybe it becomes part of the cell membrane itself. It just depends on what we need to do with that. Okay. So the Golgi is where the proteins are going to be modified. Usually this means that that little sugar chain is going to be modified as it passes through the Golgi apparatus. makes the oligosaccharide unique to do a particular job, but they all start out the same when they are in the ER, the little sugar add-ons. Okay, so again, we can kind of see this ER, we've got protein, it pinches off, the transport vesicle is going to take it and fuse onto the Golgi apparatus, spit the proteins in there where they will get modified. This is not showing the little sugar chain that would also be added at that time. Just be aware that the Golgi also will sort products by tagging them with little phosphate groups and other things that will help sort the different proteins to where they're supposed to go. Okay, so let me make sure sound is off. All right, so making proteins, we're going to start in the nucleus, we're going to make RNA, and the RNA is going to leave. The RNA is going to perhaps bump into some ribosomes sitting on the rough endoplasmic reticulum. It might go to a free ribosome, but we're going to not focus on that. We're going to focus on these on the rough ER. Okay, so perhaps this mRNA joins up with that ribosome, and that ribosome makes some protein. And that protein moves through the rough ER. It probably gets a little oligosaccharide added to it, and then it is pinched off in a little vesicle. Where is that vesicle going to go? It's going to go to the Golgi apparatus, and that Golgi apparatus will modify further that protein, usually by modifying the little carbohydrate that got stuck to it in the ER. And now we see this modified protein here, and then that little protein will get pinched off one more time into a vesicle and sent out to the bloodstream. Okay, and so this shows that protein secreted and on its way. But it could become part of the cell membrane too. We need to know what the job of the protein is. It could go anywhere. Okay, so all of this is called the endomembrane system. The endomembrane system. We've already talked about all of these except for the lysosomes. We'll talk about those later. We have not really talked about vacuoles a lot. We've talked about, however, vesicles. Vacuoles are very, very similar to vesicles. Okay. All right, different picture, same idea. If that helps you out, you can pause the screen and take a look at that as well. What I want you to see in these last couple slides is how similar animal cells are to plant cells. All those endomembrane organelles are in there. They are the same. ER, Golgi, everything is in both plant and animal cells. All right, that's it. Thanks for watching. See you next time.